Hi friends, so today I'm going to talk about the gluconeogenesis. So, by definition, you can say that the gluconeogenesis is a process of uh, generating glucose from non-carbohydrate source. In simple terms, we say that gluconeogenesis is a process by which the glucose is generated from non-carbohydrate source. Well, now we have two opposite pathways, gluconeogenesis and glycolysis. So gluconeogenesis leads to synthesis of glucose and glycolysis leads to degradation of the glucose. So these two uh, are the opposite pathway. But all the reversible steps of glycolysis and gluconeogenesis are same, but irreversible steps are different. That means these two are opposite pathways. However, all the steps of these two pathways are not similar. So the reversible steps of the glycolysis and the reversible steps of the gluconeogenesis is the same, but the irreversible steps are different. So there are three steps in glycolysis and gluconeogenesis. They are different. That means they are catalyzed by different enzymes. So this is the first step where the variation occurs. Now this is the phosphoenol pyruvate converts to pyruvate. This is the last step of the glycolysis, and this step is carried out by pyruvate kinase. But in case of gluconeogenesis, the pyruvate converts back to the phosphoenol pyruvate. So in this case, in the glycolysis, it is an irreversible step. So in this case, this pyruvate converts back to the this phosphoenol pyruvate by different enzymes, and it occurs in two steps. In the first step, the pyruvate converts to oxaloacetate with the help of the enzyme called pyruvate carboxylase. So this leads to incorporation of a carbon dioxide molecule, and then this leads to formation of oxaloacetic acid, and so it utilizes one ATP, and then the oxaloacetate now converts to phosphoenol pyruvate. So here there is a utilization of two molecules of GTP for two molecules of pyruvate to two molecules of phosphoenol pyruvate. So two molecules of GTP are utilized, uh, and the enzyme is phosphoenol pyruvate carboxykinase. This leads to formation of phosphoenol pyruvate. Now in this case, this particular reaction of glycolysis occurs in cytoplasm, but the conversion of pyruvate to oxaloacetate it occurs in the mitochondria, and oxaloacetic acetic acid converts to phosphoenol pyruvate. This also occurs in mitochondria. So the region where it occurs is different. So as I've already explained, this step occurs in mitochondria, and in glycolysis it occurs in cytoplasm. So sometimes not the pyruvate but the lactate also can act as a precursor for glucose. So lactate converts to pyruvate by the enzyme lactate dehydrogenase, and then this pyruvate enters the mitochondrial matrix. So now as we have discussed earlier, so pyruvate carboxylase. So here the carbon dioxide is incorporated to make it oxaloacetate, and then the phosphoenol pyruvate carboxykinase, this again releases the carbon dioxide making the phosphoenol pyruvate, and it goes out of the mitochondria, and then to carry out the rest steps of the gluconeogenesis. Now the second step where the variation occurs. So now in this step of glycolysis, the fructose 6 phosphate converts to fructose 1,6 bisphosphate. So but the enzyme is phosphofructokinase 1. Now the same the for this in the gluconeogenesis, this fructose 1,6 bisphosphate converts to fructose 6 phosphate, but the enzyme fructose 1,6 bisphosphatase. So here one thing to be noted regarding the enzymes used here, the kinase means the enzyme which adds the phosphate group. So here fructose 6 phosphate has got only one phosphate group, and then when you added the phosphate group it became fructose 1,6 bisphosphate. And in this case the phosphate group is removed, fructose 1,6 base phosphate to fructose 6 phosphate. So now the removal of the phosphate is done by the family of the enzymes called phosphatase. That's why the name is fructose 1,6 base phosphatase. So this is the second step where these two pathways differ. And then this is the first step of glycolysis. Glucose converts to glucose 6 phosphate with the enzyme hexokinase. And now this is the last step of the gluconeogenesis, where the glucose 6 phosphate converts to glucose at the removal of the phosphate. So as I've already explained, glucose 6 phosphatase, which removes the phosphate group. So these are the different amino acids which can be converted to pyruvate. So we call them gluconeogenic amino acids. Alanine, cysteine, glycine, serine, threonine, tryptophan, all of them they can convert to pyruvate. So there are some amino acids which convert to the TCA cycle intermediates. Just like these group of amino acids, they convert to pyruvate. Some amino acids are there, they convert to intermediates of TCA cycle. Say for example, the alanine, glutamate, histidine, proline, these amino acids, they convert to alpha ketoglutarate, which is an intermediate of the TCA cycle. So here you can say that this is arginine, glutamine, histidine, proline. These amino acids, they convert to alpha ketoglutarate of the TCA cycle, and then this cycle goes on and it makes the oxaloacetate. So oxaloacetate now converts to phosphoenol pyruvate to make the glucose. So it is the phosphoenol pyruvate, and then this will further go ahead in the pathway of the gluconeogenesis to make the glucose. So similarly, there are some, these four amino acids, they can convert to succinyl coenzyme A, which is also a part of TCA cycle. So this is the succinyl coenzyme A. So these four amino acids convert to succinyl coenzyme A, which convert to this oxaloacetate. Then this will further convert to phosphoenol pyruvate. This is occurring in the mitochondria. This will go out of the mitochondria and convert to glucose, as mentioned earlier. So these are the amino acids which convert to fumaric acid, the fumarate in the TCA cycle. So same thing, these amino acids, they will convert to fumarate, then malate, then oxaloacetate, then this will be channeled to make the phosphoenol pyruvate by the enzyme called phosphoenol pyruvate carboxykinase we have already discussed. Then this will make the glucose. So these are the gluconeogenic amino acids. So pyruvate, so this group of amino acids, they form pyruvate. This group of amino acids form alpha ketoglutarate. This group of amino acids, they form fumarate. And this group of amino acids, they form the succinyl coenzyme A. So they all lead to formation of gluconeogenic amino acid, this glucose. So that is why they're also called, in some books you find glucogenic amino acids. The same thing only. So you can take the screenshot of this table for your record. So thank you very much for watching this video. See you again.